This over text. I said, well, your volume was just about the place. I mean, coordinator. All right, everyone, we're going to go ahead and get started with our REU presentations. I want to thank all of you for being here in person and online. So tonight is the accumulation of 8 weeks of hard work by 4 of our students. We call them research experience for undergrads. They've been up here since the second week of June, right? When all the 4-week classes started. They spend every day, all day doing research, which you'll hear about today. And I would encourage all of you to look up our REU program for next year, okay, if you're interested. So the only qualification, you have to be an undergrad and still going to undergrad in the fall to be considered. We're open to OSU and non-OSU students. It's a paid position, so technically they are currently Ohio State employees until Saturday. But also, you know, our REU program and many other similar programs are really competitive. We had, I forget the exact number this year, but we get somewhere between 140 to 200 applications for 4 spots. Okay, and when we are reviewing cover letters and resumes, we learn how to weed out the bad ones really quick. Okay, so some of the things we look for are experiences that are outside the classroom. Even though you're taking classes here at Stone Lab, this is not a main campus class, right? You weren't told to be here. You came up on your own will because you're interested in it. So on your resume, make sure you went to Stone Lab, you took classes and X, Y, and Z, and you learned different skills. Okay, make sure you put on your resume or your cover letters your skills. Okay, we are much more interested in outside of classroom stuff than we are inside of classroom, and we're also more interested in stuff that is science, STEM based. If you worked at a zoo, tell us what you did at the zoo. That's much better than telling us you work at Whopper World. Okay, we kind of know what you do there. Okay, so make sure you be specific on your resumes and don't feel rejected when you don't get the offer, because we had a lot of outstanding candidates. And these were the top 4. But the 5th ranked candidate is, just right there. So we only have 4 spots. Many other programs might have 8 or 10 spots. A lot of people are applying. Okay? So I encourage all of you to look for more opportunities outside classroom. So with that, we're going to go ahead and get started. There'll be 4 presentations. The order is picked based on the food chain. So we're going to start at the lowest level and work our way up to the highest level, even though these things may not be directly eating each other. That's the way we're going to go. Each student will have about 12 minutes to give a presentation and a couple of minutes for questions. So we'll try to, hopefully we should be done by 7:45, 8 o'clock. Okay. Each supervisor will introduce the student, and then we'll get started. Okay, so our first student is my student, Kate Zaparaniuk, from Ohio State University, originally from Georgia. Kate is going to, probably hopefully I would change the screen, huh? Stop share. Okay, bear with me while I change slides. And I exit out. Okay, here we go. All right, so what you're going to hear from Kate is just a small subset of her actual research. Kate and I have been working together for a while on this project, looking at field work. Kate was up at Stone Lab last year as a summer, I don't know what we call it, a semester at Stone Lab student, where she took the 2 SE&R courses. Then on the other day did Stone Lab stuff. And that's where I met Kate, was when she was working in the lab. Okay. People online, bear with us. The computer is being slow. I know it's going to open the presentation several times now because I clicked it several times. There we go. Okay, so Kate's project is titled "To be Inside or Outside the Cell, an Examination of Lake Erie Microcystin Congeners." And then so come on up, Kate. Not yet. Not yet. Share. Let me share the screen. And we do that. Think of all the money we spent on IT over the winter, that this would work. Okay. Hold on, folks. Why is this not working? So this doesn't count towards your 15 minutes.
Okay.
Share. Share the screen first. We'll do that. And then I got to duplicate it. Well, it's display settings. All right, we got it. Thank you, everyone. And then I should be able to. No. Let's put that down. All right, there you go, Kate.
Yep. All right. So my name is Kate Zaparaniuk. I am Dr. Justin Chaffin's REU. And as he was saying, my project is to be inside or outside the cell, an exploration of Lake Erie Microcystin congeners. So harmful algal blooms, they've been occurring annually in the western basin of Lake Erie since the 1990s. You've maybe seen them on the news, or maybe you've seen a warning sign such as this at a beach near you, or maybe you've heard of the Toledo do not drink advisory that happened back in 2014. These harmful algal blooms are harmful to both human health and the environmental health, and exposure can cause nausea, skin irritation, and lung inflammation. So let's take a closer look at harmful algal blooms. They consist of a type of algae known as blue-green algae or cyanobacteria. Within the western basin of Lake Erie, it's mainly consisting of Microcystis aeruginosa, which disrupts the lake's natural food web and is a symptom of eutrophication or excess nutrients in the lake. So the algae Microcystis produces a toxin known as microcystin. This toxin is harmful to both human health and the environment. It causes both internal organ damage and can affect people with underlying conditions. Okay. So microcystin can occur in multiple different types based on the placement of amino acids. So these different types are known as congeners. So here, these amino acids we see are leucine and arginine. They combine to create microcystin LR. There are 4 different congeners that are seen most frequently in Lake Erie, which are microcystin LR, RR, LA, and YR. Out of these 4 congeners, microcystin LA is the most toxic. These congeners are very nitrogen dependent, so they require excess nitrogen in the environment to be created. So these microcystins can occur both within the algal cell and outside of the cell, known as intracellular and extracellular. The extracellular microcystin is harder to remove from water treatment, as the intracellular version can easily be filtered out just with a filter, while the extracellular can persist. It also has the potential to aerosolize in its extracellular form, where it can then be inhaled. And extracellular version of the microcystin is a bit more ubiquitous to all the organisms it affects in the lake, as the intracellular version affects organisms when they physically consume the algal cell or consume an organism with the toxin in it, while as the extracellular version is just in the water. And in the extracellular version, it can also bind to sediments more readily, and then these sediments can then persist further past peak bloom. So how does the toxin get into its extracellular form? This is mainly just through any sort of like natural cell death. And when the cell dies, the once intracellular toxin then is turned into extracellular form. And from one strain of Microcystis, there can be multiple different microcystin congeners created. And once the microcystin is extracellular, it can be consumed by heterotrophic bacteria, or basically bacteria that will consume organic matter. You can see here that this is a Microcystis colony, and all around it, you can see the heterotrophic bacteria that's associated with that colony. So because of these different congeners and their different abundance and toxicity, and because of the unique threats that the extracellular version poses, we are kind of going to take a quick look into past data to see if there's any sort of dynamic in the intracellular versus extracellular congener makeup. So we use data collected from Stone Lab in past years, and they were collected across the Western Basin, as seen in this map over here. And I just took a look at the different congener profiles in the intracellular compared to the extracellular microcystin. So this is taking a sample point just from 2018, right near peak bloom. You can see that out of the total microcystin, only about 3% of it is extracellular, and the majority of it is intracellular. Taking a closer look at the intracellular portion, you can see it's broken down into these different congeners, where microcystin LR is consisting of the most of it. Now, looking at the extracellular version, we can see that there's a similar breakdown. If we compare these two, we can see microcystin RR is higher in the intracellular version and lower in extracellular, while YR is increasing in the extracellular version. So because these two bar graphs are different, we can see that there's some sort of preferential degradation happening. If there was no degradation, then these two bar graphs would be exactly the same. So looking at across the years I looked at, we can see that the microcystin congener makeup is shifting by month, and it's also different intracellular compared to extracellular. So that leads me to my research question of, is there preferential degradation of these congeners, and what sort of external degradation factors are kind of at play here? So my hypothesis is that there will be preferential degradation of specific congeners, and also that the congeners are going to degrade at different rates based on different external factors. So what are these external factors? So what naturally degrades extracellular microcystin? We can see two different types of bacteria. There's the colony-associated bacteria, where it's around the Microcystis colony, and then there's just free-living bacteria already in the lake water. There's also the binding to sediment, photodegradation, and then binding to anything else in the lake. So this leads to my methods and my experiment. My very first step was actually culturing Microcystis itself, where I took 3 different strains of Microcystis and I combined them in order to get a full congener profile with the 4 congeners I was looking at: LA, LR, YR, and RR. The toxin, the extracellular form of this toxin was then extracted by freezing and thawing the algae strain so that I could get just the toxin itself. I then collected lake water from Lake Erie the morning of my trials, and I created these different treatments to kind of replicate the different degradation mechanisms found. So my first treatment was just raw lake water, which would expose to all sorts of natural degradation from the lake. I then have a filtered sample that would remove just the colony-associated bacteria. And then I had a treatment that was the water that was then sterilized with formaldehyde, so it would kill any bacteria, but it would leave sediment suspended. And then my last sample, I was fully filtered as much as I could, and it would remove all sorts of bacteria and all sediments. Once I created these treatments, they were then left off the docks at Gibraltar so that they could be exposed to natural fluctuations in the lake, so natural temperature fluctuations and natural sunlight fluctuations. I took samples, an initial sample before putting them in the lake water, and then I took a sample 6 hours later, 12 hours, 24, and 48. When I took these samples, I was passing them through a filter that would remove any sort of sediments or any sort of cell-bounded microcystin. These samples were then sent to Wayne State, who would then process them and determine what the congener profiles are. So we get to my results. So this graph is comparing the initial to the final microcystin makeup per treatment. And you can see that there is a difference in the microcystin makeup initial compared to final across all the treatments. Looking closer specifically at microcystin RR, you can see that it is decreasing comparing initial to final. So looking a bit closer at microcystin RR, we had to then calculate these degradation rates. So I took a plot like this and I found the change in concentration over time per bottle. And then I averaged that by treatment to find the overall degradation rate per treatment per congener. And if you can tell from this graph, the formaldehyde treatment that I used actually had an impact on the analysis because you can see it's much lower down compared to the other points. And there was this kind of interaction it had when we were analyzing the data that caused a bit of an error. So this is looking at just the raw lake water treatment degradation rates across each congener. And you can see microcystin RR looks like it's being degraded faster. So we then ran a one-way ANOVA test so that we could see if this difference was significant, and it was. So this is showing microcystin RR is being degraded faster. And this, again, this is the treatment of the raw lake water, so it's being exposed to all sorts of natural degradation types in the lake. This is showing across each of my treatments, and we can see that still microcystin RR across each treatment is still being degraded faster, but we want to isolate the mechanisms. So I use my different treatments to be able to isolate my three mechanisms I was looking at, which is the free living bacteria degradation, colony associated bacteria degradation, and then my abiotic sample, which was that filtered sample that would filter out the sediments and the bacteria. And across these, you can see that microcystin RR is still being degraded faster. We ran a two-way ANOVA, and by congener type, the difference was still significant where RR is being degraded faster. We also then looked at the different degradation types to see if they played a factor, and there was not significance with that. So looking at my data, we see that RR does have the highest degradation rate, which can lead to the conclusion that it is being preferentially degraded. It is important to note that out of the 4 congeners I looked at, RR is the least toxic. So because RR is being degraded faster, that's leaving these other congeners that are more toxic in the environment for longer, such as microcystin LA, which is the most toxic of the 4. We also saw the most degradation was from the free living bacteria, and the least was from that colony associated bacteria. This is likely because I ran my experiment in early July, and this is pre-bloom. So there weren't a lot of microcystis colonies in the lake in general. So there wasn't that as much colony associated bacteria then. There's also a potential for interaction between different degradation types within, say, like my raw lake water sample has multiple different degradation types all competing for the microcystin. Whereas once I start removing these different degradation types, it could increase degradation rates of the remaining mechanisms. So this could mean on my calculations, when I was calculating the rate based on mechanisms, those numbers could be slightly skewed. And lastly, we could not use the treatment with formaldehyde. So we could not figure out what the binding to sediment difference in degradation was. So we could not calculate sediment binding. So in the future, I would like to repeat the same experiment later in August, actually near peak bloom, so we can actually get that colony associated bacteria and hopefully get higher degradation rates in general. And I also would like to do some further testing with the formaldehyde treatment so I could get that sterile treatment to get a better understanding on how sediment binding is impacting the microcystin. So thank you to everyone who made my project possible, especially my mentor, Dr. Justin Chaffin, and Judy and her team at Wayne State, Kim from the University of Michigan, Kelly, Catherine, Josie, and Kira in the Stone Lab Water Quality Lab, my fellow REUs and students, and all of our funding. Here are my references. Thank you.
Good job. We have time for a couple questions while we switch the slides.
What are some other ways that you can try and isolate just sediments instead of the
bacteria that don't evolve. Okay, I'm glad you asked that because we actually ran a couple different trials on that because it is pretty difficult to get raw lake water and then fully sterilize it without disrupting the sediments. So we tried to use the autoclave, which is like what we use to sterilize equipment in the lab. And while that did sterilize our water, it caused all of the sediments to fall out. We also tried microwaving, which had the same result. We also used penicillin, actually, which unfortunately didn't kill as much of the bacteria as we hoped. We think that the formaldehyde, it's not necessarily like the fact that it's influencing the microcystin. We think it's on the analysis side more like because it's like with the machine we use to like isolate the different congeners and find the concentrations, the formaldehyde could be influencing that. So it's more just like running some tests on that to try and adjust it. So yeah. Mm-hmm.
Question from Dr. Kane online is, what was the most difficult part of the project?
Probably, let me think. There were a couple of hiccups we had. Probably was trying to figure out what the best way to isolate the oh, you know what? Actually, I was waiting for my data. My data took unfortunately a little bit longer than I was anticipating to come back. And that was a little bit rough, especially because we had the potential to save some of our samples to run a couple additional tests here, but we kind of wanted to get our data off as fast as we could. So we didn't really think much about that. And it was just that waiting for my data, just, yeah. Yep. Yes.
Do you have any sense of how long microsystems are persistent when bound to sediment, and if that has any implications for human interest, for human health concerns?
I've read a few articles about it. There's kind of some conflicting messages I've heard. But it definitely is persisting past peak bloom. And well, you can actually read like how much there was an algal bloom just with like past records from the sediment, but it's not necessarily leaving the toxin in there. But I think the one study I referenced, it was about a month past peak bloom that they were able to find it in like detectable amounts in the sediment. So I'm not really sure with like human impact what that would say.
Would that change with different bacterial activity, different exposures?
Yes, it definitely does. Yes, and then like where in the lake you're looking at too. Yeah.
Good job.
Thank you.
I'll ask Zach to introduce the next. All righty. I'm going to introduce Sydney Knight here in a second, but I also just wanted to say kind of how I met Sydney. Sydney actually was really interested in marine science, so probably many students in this room are really interested in marine science. So you might think coral reefs and clear water. The first day of sampling, I took Sydney to the Maumee River where the visibility was 0, and actually we were trying to go up this pond because we wanted to find this species called a giant floater. And it was like, if you've ever seen Shawshank Redemption, you're trudging through the mud and the sewage, right? That was our kind of first experience, okay? Then we didn't find any mussels there. So our second option was the Maumee River, okay? And it was a little bit deep that day, not unsafe. But the visibility was basically 0 on our mask. And so Sydney, trooper that she is, she's swimming, holding her breath, swimming underwater, feeling against the muck and the sticks, and hopefully there's no snapping turtles. And so that kind of grit and toughness, I think, has carried Sydney throughout the summer. As many of you have done with your own classes, your own little class experiments and your REU experiments, you're going to experience failure at some points, right? Or uncertainty, and maybe have to pivot your project. And Sydney has done an excellent job at that, kind of tweaking little things, but also preparing really well this summer. So I'm really excited for her to talk about her project. And without further ado, take it away, Sydney.
All right, thank you, Dr. Steffensmeyer, for that wonderful introduction, as well as the other people that have helped me out so far. Today, we're going to be talking about mussels. Some of you may not know what that is, so where we're going to start is what are mussels? A little bit of background information so that we're all on the same page. And then following a similar format to Kate's presentation, we'll go through my guiding question, my methods, my findings, and then a short discussion at the end. So getting right into it with our background, what are freshwater mussels? The class, Unioida, has 298 species here in North America, and Ohio has 60 of those species. This picture up here shows some of the differences between the species that you can find here in Ohio. They can look really different from each other. Some are larger, some are thicker, some are thinner. They also bury into the sediment of the water area and the gravel that they're in. And overall, we have very limited knowledge on their feeding regimes. So what they eat, how fast, what they like to eat, what they can, etc. And 60% of native freshwater mussels are considered endangered or threatened, with 12% being presumed extinct at this point. So they're in a massive decline. This could be due to pollution in the water, sedimentation, outcompeting by invasive species, and other factors. So it's really important that we get a better understanding on how they are functioning in the river. So the two species that we looked at in my study were the white heel splitter and the maple leaf. And as I mentioned, outcompeting by invasive species, we're going to go into the two species and two taxa that I looked at throughout my study, starting with Dracaena. These are your zebra quagga mussels that you may be familiar with, and they were introduced off of ballast water from the Black Sea over here into the Great Lakes system. And there's also a lot of debate on what they're eating. So some people have found evidence that they are selectively choosing to eat cyanobacteria. Others have found that they're feeding on everything but that. So there's no one answer yet in the literature. Our next taxa is Corbicula. These are also known as Asian clams. So they are not mussels, but they are bivalves, so similarly related. They are native to Eastern and Southern Asia, and similar to our Dracaena category and even our native mussels, we see debates on what they're choosing to eat. So how do mussels eat? This here is a white heel splitter shell. And if we were to take a look on the inside, you would see something like this. They have an in-current siphon, which is the one down here, and the water flows in through that siphon and then flows over their gills and is taken into their digestive system, which includes their stomach and their intestines, and then is essentially thrown out or spit out through their excurrent siphon, this siphon here. So my main question looks at how do freshwater bivalves change the composition of algal communities? Splitting that into two more questions. First being, do non-native taxa feed faster than native species? And secondly, is there any selective feeding in our study taxa? So I hypothesized that our native or non-native bivalves are going to be feeding faster on algal communities than our native bivalves. Evidence of outcompeting that I've seen in the literature and more. My second hypothesis is that our two native species, the maple leaf and the white heel splitter, will non-selectively feed between green algae, cyanobacteria, and diatoms, our three algal groups. Lastly, our two invasive taxa will selectively feed against cyanobacteria, meaning that we'd see them preferentially uptaking green algae and diatoms. So how did I actually do this? Over in our lab, I had 15 10-gallon tanks, as seen in that bottom right-hand photo. This model will be scaled down just to 5 so that we don't crowd that whole screen up. And we had 3 tanks with white heel splitter, 3 tanks with maple leaf, our native species, 3 control tanks, 3 Dracaena tanks, and 3 Corbicula tanks. And the first step to this experiment was mixing our algae cultures. So we had a green algae culture, a cyanobacteria culture, and then a diatom culture. And we combined those into one big jug, as seen on the bottom left, in order to make sure that they were all put together and starting at the same concentration. From there, we did some trial and error, putting a little bit of one culture into a tank, and then checking to see how much algae we had put in that tank. We repeated this for all cultures until we hit the level and the amount of algae that we were hoping to have in all tanks. This was trying to be standardized so that all 3 cultures started at the same place. Then we added our mussels into our tanks, and they got a 20-minute acclimation period from the time that they left their stock tank into their new home for the next 24 hours. They were in here for 20 minutes, and then I also conducted a visual check to make sure that their siphons were open and they were exhibiting normal feeding behaviors. After that, I began collecting fluoroprobe samples every 30 minutes for the first 2 hours. This was a lot of work, so we had to be very focused. I had a lot of help in the lab trying to run samples back and forth. We also continued to collect samples a little less intensely at 4 hours, 6 hours, and 24 hours. So this fluoroprobe machine is what's on the screen right here, and it uses different LEDs with different wavelengths in order to kind of shoot through our water sample. And then there's a sensor on the other side that gives us a numeric value for these different algal groups in micrograms per liter. Once we had collected all of this data, we began our data analysis. I'm now very friendly with R, and we ran a linear mixed effect model where our response variables were those algal groups, and then our total algae group consisted of the sum of those 3 groups. We had fixed effects of taxa, time, initial algae, and biomass, and we're able to use tank number as a random effect, since whenever we put species into a tank, it was randomly generated which species, which treatment would go into that tank. From this model, we then ran estimated marginal means, which compared the different treatment and taxa groups while controlling for the biomass or the weight of the individual and our initial algal concentrations, since there was some variability between our 4 trials. Significance for this was assessed at an alpha value of 0.05. So what did I end up finding? We're going to walk through this messy graph, and I'll break it down a little bit so that it may be easier to digest. Starting off with that super intense sampling between 0 hours and 2 hours, there's a lot of noise up there, and we really didn't see differences between our different groups in those first 2 hours. Between our first 6 hours, though, we do see this steep decline in most of our groups before it plateaus out to a bit flatter of a slope. I also want to point out our Dracaena group, which are those invasive zebra quagga mussels, are filtering much faster and rapidly declining quicker than our control in black, as well as quicker than our maple leaf, one of our native species. The Corbicula and the white heel splitter tend to hang out. Those lines overlap a lot in the beginning and have some separation, but no significance. And then our maple leaf remains pretty close to our control throughout the entire experiment. The shaded regions on this graph are confidence intervals of 95%. This is for our green algae. And we have a similar pattern in our cyanobacteria. The plateau is a little bit less evident in our cyanobacteria, but lots of noise at the beginning, decline, and then a bit of an evening off, with our Dracaena group still being the quickest. For our diatoms, you can see that our starting values are much lower. There is a similar pattern of a steeper decline to 6 hours and then a plateau. However, in this one, we actually have this blue white heel splitter coming out as the fastest filterer, taking up more diatoms than others. Again, may not be significant in the beginning, but we do kind of start to see that trend emerge. And then our total algae, which is the sum of all 3 of those, follows very, very similar, if not the exact same pattern. So I said I wanted to see how fast they were filtering though. So here's where we get our rates, which are modeled rates from our models in R. And we see one main pattern, which is that our white heel splitter, our Dracaena, and our Corbicula are selectively feeding for green algae above all else. We also see in our Dracaena that they are selectively feeding for cyanobacteria over diatoms. In our maple leaf, one of our native species, we don't see any significant selective feeding at all. And then in our control, we see a rate, and it's higher in diatoms, which could be due to sinking and not being able to suspend the particles as well. So coming back to those hypotheses that I started with, we see that for green algae and cyanobacteria, as I just mentioned, our Dracaena, or our zebra mussel up here at the top of the podium, is significantly filtering faster than all other taxa. We also can see that our other invasive taxa, Corbicula, is filtering diatoms significantly faster than our maple leaf, here in 3rd place. And our corbicula show no significant differences between the white heel splitter, hence their tie in second. This is partially supporting this hypothesis. Moving on to, are our native species selectively feeding or not? As I mentioned, our maple leaf do not show any selective feeding patterns. However, there is evidence to support the white heel splitter selectively feeding for green algae and having no preference between cyanobacteria and diatoms. Lastly, looking at our invasive taxa, we see that both show selective feeding for green algae and do not show selective feeding on diatoms, partially supporting this hypothesis. So what does this information actually mean? How can it be applied? What does it matter to you guys? This allows us to have a better understanding of bivalve algae dynamics in streams and lakes and watersheds, and also its influence on algae blooms to see if they're choosing one over another, which could take away competitors in the algal community. We also see that it's important to focus on species by species differences in uniotid species instead of clumping them all together by seeing that our maple leaves aren't selectively feeding, but our white heel splitters are. They're choosing to have green algae over everything else. And this should be accounted for in the future. Another reason that this information can help us out is better understanding feeding regimes for native captive mussels in order for restoration efforts. So if we have a breeding program and we're trying to raise them past the juvenile stage through adult, we're trying to do that the most efficiently. We need to know what they want to eat, how fast they're doing that, to be able to provide them with the nutrition that they need. In the future, I also collected nutrient data for dissolved inorganic nutrients, which was collected at our 0, 2, 4, 6, and 24 hours. So analyzing that and seeing if there is this difference between non-native and native species, as well as being able to use this data and extrapolate it out and see historic versus contemporary mussel populations and put a number on how they are affecting the water column and at what rate. Another way that we can continue to look into this is looking at how mussels and bivalves feed on other types of particles. They're just going through the water column, so there's a bunch of stuff in there, like zooplankton, or even aquatic fungi that they could be uptaking and preferentially selecting for instead of these algal groups. And lastly, seeing how this selective feeding changes in a natural system where there could be competition or niche partitioning between species. Thank you to everyone who made this project possible, including my mentor, Dr. Zach Steffensmeyer, Dr. Justin Chaffin, our research coordinator, Dr. Eva Rosnier for all the feedback that she's given us, to the entire Stone Lab Water Quality Lab, including Kira, Catherine, Kelly, and Josie. Your guys' patience and time that you put into this project with me is crucial. Thank you to Kim from the University of Michigan, who provided us with our Microcystis culture. Thank you to our funding from Ohio Sea Grant and from Ohio State University and Stone Lab for making this experience possible for me. And lastly, to everyone else who made this summer exceptionally memorable. Here are my references. Thank you.
We have time for questions.
Good job. What are some of the challenges that you faced during your time doing this project?
Yeah, so as invasive as our Dracaena category is, they must have known they were being experimented on because they died right before a few of my trials, which meant that I had to go collect more. We did not have to go through as tumultuous as a process as Zach explained with our native mussels. Those guys were able to just grab off the back of Gibraltar. But nonetheless, that was pretty frustrating having them die in multiple rounds. That would probably be my number one. In the way back?
Yeah, so you're the muscles down by 60%.
Not that I know of. We have another paper from 98 that essentially says the exact same information, but groups that together as 72% to include the extinct population kind of in that. So this is something that is still happening. They're still in rapid decline. and our limited knowledge doesn't really help any efforts to boost populations. Yep. Here in the middle. Yeah.
This is controlled earlier in the spread by environment was so stunning also. Further how you read the graphs.
Yes. Is there one in particular?
Just
It's okay. I'll explain.
the conclusion that the maple leaf was not selective for feeding.
Yeah.
Even though they were feeding on things that was rates.
Yeah. So in that graph, our bar graphs came down and then we had error bars. So the overlap of all three of those, where we see them really close together and their error bars are overlapping, is what shows us that there's no selective feeding to any significant level that we see, compared to those ones that had asterisks on the bottom with the green algae. The error bars on either side don't touch a different algal group. So that's how we know that they're statistically significant.
And then there's two questions online. The second one probably be easier.
Okay.
So have you thought about the possibility of expanding trials past 1 day?
Great question. So they more than depleted the algae that they had in their tanks after a day. So we ended it after 24 hours. There would be slight mounts of cyanobacteria for them to eat through and could put them in like a starvation period or abnormal feeding. So that's why we ended it when we did.
And then the second one is, do you think they're spitting anything out?
They definitely could be. One thing that mussels do is they have pseudofeces, which is kind of their way of spitting things out. So there's a couple of different stages that they can be sorting these particles. We looked at how it changed in the water column, but as they pass over their gills and go into their digestive system, there could also be sorting happening there, which is then rejected in their pseudofeces or feces that we didn't account for.
Yeah. All right, I'll ask Steve to come up and introduce Jihae.
Hi everyone, Steve McMurray from Old Woman Creek. This year was the first year that we, I guess, had a co-relationship between Stone Lab and Old Woman Creek, where I had the pleasure of working with Jihae Dick from Ashland University. She actually stayed on the island, Stone Lab, but she did her field work at Old Woman Creek. I'm an invertebrate biologist, so I was kind of taken aback when she said she wanted to work with fish, but we spent a lot of time on a boat over the summer sampling fish, filtering water. What she's going to talk about today is just a small portion of her work. Go ahead and take it away, Jihae.
Thank you, Dr. McMurray, for that wonderful introduction, and thank you for everyone who came here today to watch our presentations. I want you all to imagine you're handed a cold case. This mystery has data going back decades, but there's still a couple questions left unanswered. Now, the interesting thing about this case is that the clues are actually fish, and they have a lot to tell us about the scene at hand. Allow me to introduce you to the cold case of Old Woman Creek, what 40 years of fish surveys reveal about estuarine biodiversity. Now, first things first, I want to outline the question at hand, if it would like to be revealed. There we go. Yeah, we're looking at how the fish community of Old Woman Creek has changed from historic records in the past up until the present, and we want to know what this can tell us about the health of the estuary. Now, this includes not only looking at the current sample of fish that's in the estuary at this very moment, but also looking at historical assemblages and identifying general shifts in the community overall. But yeah, just a roadmap of what we're going to talk about today. First is the mystery. I'm going to introduce you to what the preserve is and what an estuary is. After that, going into our methods, which are primarily electrovision, which I'll reveal a little bit later. And then the clues, which are our results, the case file, which is our previous studies in the historical record. And then after that, we're going to use all this information to solve the mystery, and discuss what it means for the estuary. But yes, welcome to Old Woman Creek National Estuary and Research Reserve. This is off a little bit in the distance, which I made the drive every week or so, but it's in Huron, Ohio. It's about an hour away, and it's one of the last untouched natural estuaries left on the Lake Erie shoreline, and it's a great natural laboratory to study fish. This is Old Woman Creek. It's 570 acres of freshwater estuary habitat. It's a regionally important fish spawning, nursery, and feeding habitat, and it's got a lot more to it than just the estuary. It's got marshes, swamp forests, open water, prairie, and cove habitat. We're mainly looking at where the fish are living for this study, but highly recommend visiting if you haven't already. First things about the estuary is that we should answer the question, what is an estuary? This is where a river meets a larger body of water and creates a dynamic transition zone between two chemically different sources. So we'll see differences in temperature, dissolved oxygen, pH, and conductivity when, say, a river meets Lake Erie or the ocean, but it creates that biodiverse and dynamic environment where we can see these unique trends in the ecosystems. Now, one thing I'd like to point out about Old Woman Creek is that it has a barrier beach. Now, this is a natural sandy beach that opens and closes on its own, depending on the time of year. This helps create that dynamic water chemistry in the estuary, allowing lake water to move in at some points, and sometimes when it closes, it is dominated mainly by the river Old Woman Creek itself. So this will have an effect on the fish we see in terms of their abundance, and also who's there in terms of species. But just to note that the barrier beach was open throughout the survey, and it only just recently closed. But yes, going into our methods, I'll talk a little bit about electrofishing, how it works, how our study design is meant to explore abundance and also other measures of the fish community, and then going into results a little later. But for summer 2026, we conducted a fish survey across 4 different sampling locations. Now, 3 of these are estuary sites in Old Woman Creek, which are up here by the Lake Erie and estuary mouth. And we also had a riverine site down here by underneath a bridge. And then this is primarily showcasing like riverine creek species, and they're a much different assemblage than what we see up here by the lake. But we ran 3 trials, sampling about once a week, and then the beach was open through this study. But yeah, going through site by site. Site A is the northernmost one. It's beside the barrier beach, which is going to affect the fish we see there. And it's primarily dominated by Phragmites and then whitewater lily. A little bit upstream is Site B, which is a wooded cove. It's by the lookout next to the Research Reserve Visitor Center, and it's got American lotus and water lily. Site C is pretty similar to Site B, it's just further upstream. It's a little bit past a railroad bridge, and it's got the same type of vegetation. And then Site D is where things get interesting. This is in the creek proper. It's riverine, it's got riparian vegetation, and it's underneath Mason Road. Yeah, going into our sampling methods, electrofishing can be done either by boat or by backpack for like shallow water habitats. So in the estuary, we can drive the boat around, and then in Site D, we had to use a backpack thanks to ODNR and the Division of Wildlife. They sent out some biologists with us, so we had help to sample the creek, and then we had our own staff like catch the fish with nets. So big thanks to them for making that possible. But how does electrofishing work? Essentially, we're creating an electric field in the water using a cathode and anode. Now, whether it's a boat or a backpack, the cathode is in the back and the anode is in the front, and that creates this gradient where when a fish swims in, it's stunned, and then we are able to scoop it up, put it in like a holding area tank, and then hold it there until we're ready to ID them. Yeah, for our study design, we ran a 100-meter transect for about 10 minutes of shock time. So we mosey along either in the boat or with the backpack, and we catch and store fish as we went along until the very end. And when 10 minutes was up, we would ID everything and then measure weight and then fork length. So that's pretty much just from the head of the fish to the divot of the tail. And if it did not have a divot in the caudal fin, then we would just measure total length, which is just the length of the fish. And then one important thing to point out is that for any fish that's less than 3 centimeters, we called it young of year. So we didn't take a weight and we didn't take a specific like length measurement. But yeah, it's one thing to kind of throw out all the methods at you, and it's another thing to see it actually happen in action. So this is a small like snippet of what it looks like to electrofish in the field. Now it's definitely a lot of work. So we've been really lucky to have people from the creek like come out, either volunteers or people who are staff working there. And then having them either net or drive the boat or ID fish has been fantastic. But this is a little like snapshot of fish we've caught, what the net looks like when we're catching like fish in the river, and then a little really bad snapshot of the boat. Yeah, going into results, we want to know what the scene looks like right now. That's why we sampled all these fish for about a month. And going into it, this is our overall summary of what fish are making up the population in the estuary. Now, there's a couple things that stand out right away. We have up here our 2 dominant species, the central stone roller, and then a regular goldfish. There's not really anything else to it, but there's a couple invasives I want to point out just to get out of the way. So we have our goldfish here. In the middle, we have our common carp, and then down here we have our white perch. And the rest of these are natives. But back to these 2 guys. So making up about 18% of the relative abundance of species we caught throughout the entire survey, in the entire estuary. These are our dominant species. Now, central stone roller is primarily found in the river, and then goldfish are primarily found in the estuary. But a couple runner-ups, these are riverine sites. Riverine site like species I'm going to mention, rainbow darter, creek chub, and then these 2 are going to be estuary site species, yellow perch, and then bluegill. But for species that are a little more rare, we'll still see all of our natives like bullhead catfish, pumpkin seeds, different types of sunfish, and then a lot of minnows, a lot of different darters. Yeah, an important thing I'd like to point out is that the abundance of fish at each site was not very equivalent. So site D, which is in the river, was way more abundant with fish, and they're not all the same size. So in the estuary, we have more adult fish that are greater than 5 centimeters. We'll get our larger catches there. So like fish that are multiple kilograms, like heavy, will be in those estuary sites, and less so in the river. In the river, it's mainly young of year, which are less than 3 centimeters, and then on average between 3 and 5 centimeters long. But they are definitely showing up at the creek. But a good way to kind of take into account that abundance isn't necessarily representative of like species that are dominant, as in like larger and better able to compete. Up here we have species biomass that we captured throughout the study, and we see our goldfish friend once again. Not too surprising, they're so abundant that even if they're a little bit smaller than the other species, they are making up a dominant part of the entire biomass. And then pointing out this guy, this is our common carp. We only caught 7 common carp the entire month out of 1,400 fish, but they are making up an important part of the biomass we caught throughout the entire study. So you'll get an idea of all the other fish we caught making up this little portion, and then these guys, goldfish, common carp, and largemouth bass, making up the majority of biomass in the study. So these guys will come up again in this presentation, just as they're invasives with big impacts on the estuary. But that was a large overview of the estuary and biodiversity we find there itself. I wanted to break down site by site for you what the fish community looks like. So over here, we've got our 3 estuary sites. So as you saw with the larger, like wide scale data, the goldfish are pretty dominant, making up between 42 and 48% of the total abundance that we caught there. But we still get a good showing from native species like yellow perch, which are down here, bluegill, creek chub, largemouth bass, and then in general, our true minnow families. And then in Site D, things get a little more interesting. We see community here is dominated by central stone roller, creek chub, and rainbow darter. These are all native, typical warm water Ohio River species. And this is indicative of a pretty healthy habitat upstream. So we don't see the invasive dominated species like goldfish making their way up as much. And then taking all of that data we took very recently, this like last month, we're going to compare it a little bit to historic evidence from the previous fish surveys we've seen in the past. So we're here in 2026, and there's 4 studies that are pretty similar to the methods we've been running, but the 2 that are what our study is modeled after are the Hoffman 1985 paper and the Bramwell 2009 paper. These are both electrofishing studies, and then they're capturing the same sites in general as we are. And then these other 2 papers are just doing general like fish surveys using netting. But if you look here, you'll see in general, we start off with 46 fish species documented in this study back in the 80s, and then 22 here, 34 in 2009, 36 in 2017, and then 24 in 2026. Now, in the 80s, 22 fish species are probably documented because it's more of the river habitat and less the estuary. So just with that sampling method, it's a little harder to compare. But in general, we see a little bit of a decline in biodiversity. But in the present, we have a moderate diversity score of 1.48 on the Shannon-Wider Index. And then that's pretty typical of a community where we have goldfish dominating as an invasive species and then less of everything else in an evenness. But an important case study I want to point out that shows this trend of declining native populations is northern pike. Now, in the 1950s and earlier, they were very abundant in Old Meat Creek, so much so that ODNR used this population to stock other fishing areas all over the state. But in the 80s, they started declining and became rarer and rarer until today. They're extremely rare in the creek and pretty much are not detected very often. They were not detected in the surveys I just mentioned past 2001. But this brings us to discussions and conclusions. We're going to go over all this fish data and sum up what it means for the creek just as a preserve. This graph shows species presence throughout time in Old Woman Creek. And these guys at the top are 3 invasive species, goldfish, carp, and white perch. So you'll see that even in the 80s, they had a pretty good foothold in the estuary, and they maintained that throughout time, even after 40 years. We still see some of our natives are pretty tolerant. They're hanging on even throughout that time. And then a couple species, like the northern pike, are dropping off. Smallmouth bass and orange-spotted sunfish are 2 of them. But we see a couple of returns in our studies. So common shiner returned in 2026, or was just identified in season, and then so is golden red horse. One important stat I'd like to point out is that in the 3 estuary sites, so not the river, if we calculate the abundance relatively for goldfish in this area for this year, that's 46.2% of our catch was goldfish in the estuary. Now, if we compare this to 2017, that's 26.7, and in 2009, it's 16.7. So we see about a 10% jump every time there's been a decade in the abundance of goldfish. So this indicates that there's an increasing abundance of invasive species. And that is important because these lead to habitat alteration by the goldfish throughout the creek. So these are our friends, the common carp and the goldfish. They are both bottom feeders. They increase water turbidity when they feed by kicking up sediment and also uprooting plants. So this is very important because previous studies, such as Mackey 2017, which is one of our fish surveys, noted that vegetation presence is an important factor in biodiversity in the creek specifically. So when that disappears, we have less habitat for our native species. And then both of these species have a similar effect. So the carp are important in that they're higher, they're more representative of biomass, and then goldfish are more abundant in general. But our major takeaways are that while fish biodiversity is moderately good in the creek, the community composition is skewed towards invasives like goldfish, and then they're continuing to be very dominant in the estuary and growing in dominance. And then for the creek proper, which is Site D, the river site, this has a fish community that is typical and very healthy for a warm water Ohio stream. So there is hope there. So that's an upstream habitat. So there could be trickle-down effects from there. But overall, fish biodiversity has decreased over the past 40 years, most likely due to degrading water quality and then any changes in aquatic habitat through invasive species like goldfish. However, it's still an important refuge for fish. We see our natives still maintaining their strongholds even today. For future work, we're hoping to conduct and actually run results on this year is eDNA. This is to get a more widespread scope of what species are in the creek, because there's still species that are more cryptic and harder to detect, even with traditional electrofishing sampling. This is a good way to detect everything that's had like a trace in the estuary. But thank you to Ohio State, Sea Grant, and NOAA for making this REU possible. And special thanks to Oldham Creek for letting me run this project and supporting me with all the equipment, staffing, and then emotional support through all the research and analysis. And then thank you to the associated divisions in ODNR, especially Division of Wildlife for their backpack electrofishing help. And then once again, thank you to all the staff at the preserve for all their help coming out on the boat every day. Dr. Ben Bramwell, who gave us the eDNA supplies we needed. Dr. Scott Lynn for sharing the original data sets for the Bramwell paper we used to make our methods. ODNR's Division of Wildlife and their two fish biologists, Katie and Josh, for coming out into the river and climbing like the slopes to get down to the bridge and catching fish with us. And then thank you to OSU's videographer Clay for taking all the pictures of us, and then my REU cohort, my peers at Stone Lab.
We can take a question or two while I transition slides.
Great point. That's a really important concern for eDNA work. So if there's any like trace of a fish, like the bird you mentioned, like if a bird ate a fish and then came down to the estuary to do its business, then there would be DNA in the estuary from that. So a hard part about eDNA is that you can detect anything, and especially when you're trying to like not contaminate your sample, it's really hard not to pick up stuff when you work in a fish lab and have all that coming in. So when we do the eDNA sampling, we're outside at the site, and then we clean everything on site and then move on. But luckily, there's a pretty good record of what is in Oldham Creek species-wise. So if there's like something really wild there, we would probably be able to tell. Oh, in the back?
It's like reminds me of spot and we make claims that are used to see the amount of first grading license for example. Ecosystem which is one invasive estuary.
Okay. Two-part question, or two-part answer, sorry. So for our methods, one of the nice things is that we were replicating site selection and methods from those earliest studies. So the locations we were sampling in were determined basically by those papers. For why we didn't add another site a little bit more upstream, there is, I think, I heard from kayakers before I started that there is a log in the middle of the estuary right above site C. So that wasn't moving anytime soon. So we probably wouldn't have been able to get up there with the boat. But also when the estuary narrows into the river, it's really hard to drive a boat up there. So you'd have to hike out onto like private property.
We'll take one more.
Was there any trends or findings that didn't quite fit into this analysis that you showed us that you still found quite interesting?
Well, that's a good question. So I did abundance analysis for each site and then like mapped out which species were there. So it's more like highlights for those. So I didn't include them, but I was a big fan of site C and D for having like the 2 green sunfish we found the entire survey. And then there were, I think there was 1 black crappie out of the whole like survey in site A. That was really cool. So we had to identify that like the last day. And it was like, huh, I haven't seen this one before.
Thank you. And Harrison will introduce our last speaker.
I brought my notebook because this is my first time giving an introduction for a student doing a research project. So my name is Harrison Freed. I've had the pleasure of working with Evie Shepard on her REU project this summer. She's an EADS major, that's an Environment, Economy, Development, and Sustainability major in SUNR. And you might be asking, why would an EADS major be up here at Sun Laboratory conducting natural science research? Well, she's not doing natural science research. This is the first ever, correct me if I'm wrong, Justin, the first ever social science REU project up here at Sun Laboratory. So she's something of a pioneer in the field here up at Sun Laboratory. Evie today will talk to you all about how she studied something called systems thinking within stakeholders who are related to harmful algal bloom management here in Northwest Ohio. Systems thinking, as she'll describe, is something that requires somebody to understand the interconnections between critical components of a problem like harmful algal blooms. And to be able to do that, to think in systems, you have to appreciate those interconnections. And I think Evie embodies that appreciation in herself. You'll see that throughout the presentation. So I'm excited for you all to hear from her in a couple of minutes. The last thing I'll say is that this is something of a full circle moment. I did an REU project here myself a number of years ago, and it's exciting to see Evie take on the REU project. She's much more put together than I was at this time in my life. So I'm excited to come back to Sun Laboratory in 10 years to see her advising a REU project of her own. So, Evie?
Hello. So yeah, like Dr. Freed mentioned, I'm going to be talking about my project, which is a social science project. So we're moving up the food chain, and I was studying humans all summer. So first, I want to get into a little bit about Lake Erie management challenges. So Lake Erie is the shallowest, the southernmost, and the warmest of the Great Lakes, which means it's the most biologically active. That's good for the fishery that we have here. It gives us a great freshwater fishery, but it also means that especially the Western Basin is really susceptible to harmful algal blooms. Some factors that lead to harmful algal blooms include climate change, soil erosion, fertilizer, and other land use patterns. Thinking specifically about land use in the Ohio portion of the Western Lake Erie Basin, we can see here that it's mostly agriculture, and which is in that yellow, and then the red is urban development. So Lake Erie, especially the Western Basin, experiences a lot of sediment and nutrient runoff from agricultural fields, and then a lot of wastewater treatment discharge. So it's important to think about how the land use patterns are impacting the lake, and also how stakeholder perceptions are impacting Lake Erie. So managing harmful algal blooms can be considered what's called a wicked problem, because there are so many different stakeholders involved. There's farmers managing the land, there's people researching up here, there's people who use Lake Erie for recreation or drinking water. So there's a lot of people involved. And it's important for these stakeholder groups to work together in order to successfully manage our resource. But in order to work together, we have to kind of build that shared understanding so that we have like a common starting point. One way we can kind of build that common starting point is through systems thinking, as Dr. Freed mentioned. So systems thinking is a way of understanding the whole structure rather than just isolated parts. So individuals that can demonstrate systems thinking may be more likely to build a shared understanding in order to address a problem, especially a problem like harmful algal blooms. So we want to figure out how to measure the ability of these stakeholders to demonstrate systems thinking. And we did this using something called mental models, which are visual representations of someone's understanding of a complex system. So for example, if I'm thinking about my lunch plans, like I know I'm moving out tomorrow, so I'm going to be making my own lunch tomorrow instead of eating our lovely Stone Lab meals. So I'm thinking of what I want to eat, and something that might influence that is what I had for lunch the day before. Something else that might influence it is what ingredients I have available. And there's tons of other connections that we can keep building. Now, mental models we collected mental models of our different participants, and these are what they look like. They're pretty complex, but they're a great way to capture the socio-ecological complexities of harmful algal bloom management, specifically how our stakeholders understood them. So we captured these mental models from 3 different stakeholder groups: farmers, which are just our agricultural producers in Northwest Ohio; technical assistance providers, which are people who help farmers make decisions on their field; and then aquatic resource managers. These are people who are researching the lake and its water quality and ecosystems around it. A little bit more about these mental models that we built with them. So all of these concepts that they shared with us were put into nodes. That's what these circles are. And we drew connections between the nodes, and these connections are called edges. Now for our study, we divided our nodes into either farm or lake nodes. We categorized them differently. This is because we wanted to analyze someone's ability to think across boundaries. So anytime they made a connection between a farm and a lake concept, or the other way around. So this brings me to my first research question of how do mental models differ between stakeholder groups? So we hypothesized that the mental models for farmers will have a higher proportion of farm nodes than technical assistance providers and aquatic managers. Our second research question is what explains cross-boundary systems thinking? So we hypothesized that greater familiarity with a participant's non-profession side will lead to, is associated with greater cross-boundary systems thinking. So getting into our methods, so this was our sample size, or our sample area. It was looking at, again, those counties in Northwest Ohio in the Western Lake Erie Basin watershed. And our goal was to conduct 15 interviews with each of our population groups. So again, farmers, technical assistance providers, and aquatic managers. We started our outreach with just internet searches. So we got contact information from different websites. And then our subsequent outreach was done through snowball sampling. So we asked participants, who else can we interview, and got their contact information from them. We contacted a total of 106 people, and we were able to complete 45 interviews. Getting into the interview itself, so our study was approved by OSU IRB, and then we had 24 virtual interviews and 21 in-person ones. It was up to the participant which one they wanted to do. And we started our interview with that silly lunch example so that I could explain what a mental model was. So we were kind of on the same page, and they knew how we wanted them to verbalize their thinking. So the bulk of the interview was actually creating their mental model. So we had our board split into 2 different sides. We had on-farm systems and lake and aquatic systems. We started them on one of the sides and asked them to just share the first thing that came to mind, either when it came to land management on farms or challenges with lake and aquatic systems. And then we continued prompting them until they had nothing else to add, switched them to the other side. And then once they were done with that, we asked them to make connections across both sides. After we made the mental model, we had a brief survey, and the survey was used to create the familiarity score, which is measuring a participant's likely familiarity with their non-profession side. So for example, how well an aquatic manager knows farming in Northwest Ohio. So getting into our results. So we first wanted to know how mental models differ between stakeholder groups. So we pulled out the proportion of on-farm nodes for each mental map. And we divided that up by population group and found that this proportion differs significantly across the population groups. Specifically, farmers have the highest number of farm nodes out of their total nodes compared to technical assistance providers and managers. And then we saw the same trend with the lake side. So aquatic managers knew more about mentioned more lake systems than technical assistance providers and farmers. So getting into our second question was looking at systems thinking was one part. So in network analysis, these triangles are often representative of systems thinking. Something else that is representative of systems thinking is a higher ratio of receiver to transmitter nodes. So receiver nodes have a higher in degree and transmitter nodes have a higher out degree. Both of these are good measures of systems thinking because it shows that someone is thinking about multiple different ways that something can be influenced and they're thinking about that end goal. So we created the score called cross-boundary systems thinking. And we did this, again, we had the farm nodes and the lake nodes that we sorted out. And we counted the number of mixed edges, the number of mixed triangles, and then the ratio of receiver to transmitter nodes. So again, those green nodes are receiver nodes because they have more arrows going in, and then the pink ones are transmitter nodes because they have more arrows going out. So we wanted to compare this cross-boundary systems thinking to that familiarity score that we built using the survey. And we found not really a correlation between the two of them. There wasn't really much of a relationship, but we were curious at how each of the subparts of the cross-boundary systems thinking score were associated with the familiarity score. So we broke them up and we found a correlation between the ratio of receiver to transmitter nodes to the familiarity score. So again, that receiver to transmitter ratio shows, if it's higher, it shows more complex overall systems thinking. And we found a correlation between that, and we did find significance when we ran a linear regression. So we can say that an increase in familiarity with the other side is likely to lead to more complex overall systems thinking. We were also curious about looking at our data more qualitatively. So for this graph, it is pulling out the top 10 nodes of all of the mental models for the on-farm sides. And it's pulling them out by the number of total connections. So if a node has more connections, that means that a participant thought that it was more central to their mental map. So they're drawing more connections to it. It's more in the middle of their map. So we can see here, this is again, just looking at on-farm concepts. And we can see that farmers talked a lot about cover crop usage, on-farm water management, and soil health. These three concepts were pretty central to how they think about the system. On the other hand, aquatic managers and technical assistance providers had more general on-farm practices and nutrient management as sort of central to their system. Now, this is similarly, this is pulling out the top 10 nodes with the most connections for the lake and aquatic side. And we can see here that harmful algal blooms is pretty central to all three of our groups. So we have this concept. A lot of people are tying what they think about back to this concept. Another interesting thing to note is that technical assistance providers and farmers talked about how farmers are blamed for harmful algal blooms as a more central concept in their system, whereas aquatic managers didn't bring it up as one of the most central concepts. So getting into our discussion of just looking at how these mental models are different. So farmers are prioritizing those farm concepts. So they understand more about farm systems, so they're talking more about it and have more farm nodes. Managers are prioritizing lake concepts. They have more of those lake and aquatic system nodes. And then we also saw that technical assistance providers fell somewhere in the middle. We also saw that across all populations, greater familiarity with their non-profession side is associated with greater overall systems thinking. Not with cross-boundary systems thinking, but it still showed that they were able to think in systems thinking. There are also some noteworthy differences in those central concepts by population group. So there were differences in how people thought about the farm system and what they thought was central to it. Farmers were getting more specific on their practices, whereas managers and technical assistance providers had more general central concepts. And then additionally, there was that farmers being assigned the blame, and that was mostly thought of as central to technical assistance providers and farmers, but not managers. But we did see that all three populations strongly centralize the role of harmful algal blooms. And that's important because that can be a really great starting point of where we can build out shared understanding. It's already a concept that people tie a lot of different things into. So it's something that we can continue other education and outreach efforts on. So looking forward, we saw that technical assistance providers kind of fell in the middle of aquatic managers and farmers when it came to how they talked about this system. So they can be kind of that go-between of farmers and managers to help build that shared understanding and try and decrease some of that difference in what people thought as central to this system. There's also future research possibilities of looking at how media and social connections influence mental models surrounding harmful algal blooms. This came up a lot in our interviews. People were talking about how media influenced perceptions and talking about other people that they knew within the system. Yeah, there are my references. And I would like to thank my supervisor, Dr. Harrison Freed, and Heather Kraska, who helped us with the data collection, Dr. Justin Traffin, who's the research coordinator at Stone Lab, and then OSU, Ohio Sea Grant, and FT Stone Laboratory for this amazing program, and then everyone who participated in interviews. And then I would also like to thank my fellow REUs and students on Gibraltar. Thank you.
Time for a question or two.
Place it in their, like however they would consider what their natural flow thought is versus how they would categorize it by farm and the water separately.
I think having them, I think they naturally would have thought of them in two different sides. There were a couple people who we started them on the lake side and then kind of immediately jumped over to the farm side. So I think naturally they would have talked about what they felt most confident in first. So farmers would talk about farming first and then managers the lake side. And then with prompting, we would be able to take them to the other side. So I think the way it was set up was pretty similar to how they would have generally talked about it. Yeah.
In area familiar with it. I'm wondering if there's a, if there's any correlation between, people's ability to connect with different ideas and it could actually change their actual behavior. Is there some so there's cognitive dissonance that happens?
Yeah, we didn't look too much. Yeah, we didn't look too much into behavior. We asked a little bit about farmers' on-farm practices because we originally were curious about how their ability to think in systems might influence their conservation practices. But one thing is we didn't have incentives to provide farmers. So the farmers that we were able to interview were the ones who were really, really excited to talk about conservation. So they're the ones who are already kind of high up on the conservation scale. So we weren't really measuring any other behavioral decisions. Yeah.
Your methods, were you ever concerned about leading? Or like introducing the topic that you might lead some of the responses?
Yeah, we've definitely tried to avoid that. A lot of the prompting questions we had were very vague. It was like they would bring up a concept and we would ask, well, what affects that? And then we would just wait for them to respond. Sometimes there were we would have a lot of concepts on the map and maybe I would be like, well, I know someone else has talked about that. And so I would try not to point out those specifically, but I would be like, is there anything else that you want to connect to that we've already drawn?
You'll use this data which you've collected doing future studies or future actions, and if not, review, who do you think will use this information?
Yeah, that's a great question. I think there's further research that can be done with, or further examination of this data that we hope to do. But also I think we want this to be sort of a resource for people who are making decisions in this system. So again, those technical assistance providers who are working with farmers, if they work for OSU Extension, they're working with farmers, but they're also working with managers. Hopefully they can use this either method of drawing out mental models or this data to kind of help them bridge that gap between these two groups.
All right, we're going to wrap this up. Let's thank our speakers one more time. Thanks all of you for joining online. And for the students here, good luck.